Hello, my name is Lenura. I have been working at Columbia University this summer under the supervision of Dr. Pascal Felicius. And the topic of my discussion today is exploring motor diversity and C. elegans work. So what is a neuron? A neuron is neurons are the cells that are able to translate chemical and electrical messages throughout the body. But what exactly is a motor neuron? Motor neuron is a type of cell that directly or indirectly controls the contraction or relaxation in the muscles, leading to the movement. If you go through the anatomy of the motor neuron, it consists of three parts. It's the dendrite, this part, the axon, and the cell body. So there are several motor neurons diseases that are being caused by the malfunctioning of certain neurons. There are two types of motor neuron diseases. One is adult onset, another one is the childhood onset. Diseases such as amyotrophic lateral sclerosis and primary lateral sclerosis are considered to be adult onset. In other words, they're most likely to occur in humans at the adult stage. And diseases such as spinal muscular atrophy are most likely to occur in children. So there are several. In order to find such cure for the diseases, scientists need to really look into different types of neurons and define why each type of neuron is different and why is it unique. So there are about 500 motor neuron classes in the human body, and some include upper motor neurons, which are located in the brain, spinal lower motor neurons, and so on. So there are all 500 motor neuron classes. It is known that there are almost 120,000 neurons in the spinal cord alone in the human body. This tells us that our organism is way too complex, and it's very hard for scientists to actually look at each neuron in the body. That's why they came up with an easier technique of using simpler organisms like C. elegans. C. elegans contains 19 motor neuron classes in the whole body, and it's much simpler. Also, if you look anatomically, it's the ventral nerve cord of the C. elegans can be compared to the spinal cord of the human body. So, and the C. elegans has been used as a model organism to actually define each neuron inside of it. It was first established as a model organism by C.B. Brenner in 1963. And what exactly makes C. elegans special? Some of the features is that it's very small. It's a microscopic size. It's about 1.2 millimeters. It's very fast reproducing, and it can take about a day for it to reproduce. It has a short life cycle of about 3 days, which makes it easier for us also. Also, it can be grown and in the lab settings, and it will be still acting the same as they would in the lab. The only thing that you need for it to survive in the lab is the E. coli cultured petri dish. And it contains 305 motor neurons in the whole body, which makes it much easier for scientists to actually study it. There are two types of sexes that C. elegans has. One is the hermaphrodite, and another one is the male. The difference, something that determines the difference between the two, is the 6th chromosome of the C. elegans. If the 6th chromosome is X, then the C. elegans will turn into a hermaphrodite. And if the 6th chromosome is missing, it will result in male. In our experimentation, we mostly use the hermaphrodite because it has unique features. It has both oocytes and sperm, which allows self-fertilization happen. So we don't have to wait for crossing to happen between hermaphrodite and male. So just like in humans, C. elegans contain different subtypes of neurons. In other ways, groups of neurons, they make each motor neuron special. Some of them are listed in here. It's the SAB, DA, DB, VA. And in each group, the motor neurons are named in the following manner, as for example, the DB1, DB2, and so on. In this study, we mostly focused on the DA subgroup and mostly on the DA9 motor neuron, which is located around the tail of the cervix worm. Also, although we know that all these subgroups and subtypes and all of these motor neurons have their unique features and they are all different from each other. We have noticed that all of these, except the VC subtype, are dependent on the transcription factor called UNC3+. So that made us wonder why are these subtypes different from these subtypes, and how the subtypes in all of those who depend on transcription factor are different amongst each other. How they're different. So looking at the DA9, we asked ourselves a question: why is it so special? What makes it different? And when we compared the morphological features of DA9 compared to others, we have noticed that, for example, the motor neuron called DA3 is extended along the body, above the belly of the worm, and closer to the head, forming the shape almost like a T, like a T. But when we looked at the DA9 motor neuron, we have noticed that it extends from the belly towards the tail and back to the belly, which always forms the U shape. That already shows one difference between the DA9 compared to other DA motor neurons. And one thing is morphological difference. Another thing is different gene expression. And in this experimentation, we mostly focused on the expression of an ITR1 gene. And something that we have noticed is that in the DA9 motor neuron, with the presence of the transcription factor UNC3, the ITR1 gene and other genes are able to be expressed and work properly in all motor neurons. However, in other motor neurons, DA9 or other motor neurons that are located along the ventral nerve cord, which I earlier compared to the spinal cord in humans, although the transcription factor UNC3 is present, the gene is not able to be expressed. So something that we have hypothesized is that there's a repressor in other ventral nerve cord cholinergic neurons that does not allow the expression of a DA9 specific genes along the ventral nerve cord. So this repressor Y in this case prevents that expression of the gene. So to come up with a solution to this problem, we have thought of a chemical named ethyl mesametsulfonate, which can cause random mutations and basically make this repressor gene ineffective, which will later result in the transcription, which will later lead to the ITR1 being expressed in these genes also. So how exactly does this chemical work? The EMS that stands for, like I said, ethyl mesametsulfonate, causes random mutations in the worm and also changes the DNA nucleotide sequence. For example, if before this was your DNA sequence, after the EMS chemical will be applied, the A nucleotide base pair will change into the G. And the frequency of such mutation occurred is 1 in 2,000 nucleotides. So how does that work? The methodology of our experimentation. The generation of worms that were put into the EMS chemical solution were named the P0 generation. And we had around 10,000 worms in that generation. They were put in the EMS solution, and it's extremely toxic, so I didn't have really access to it, and my mentor was doing that part of the experiment. It could cause cancer and many other disorders, so that's why I'm not allowed to be working with that. And after this generation was being put in this chemical generation, we have put 2 worms per petri dish, and this will be called the F1 generation. And after that, this generation was incubated at different temperatures according to which day we needed them to be ready. And after it laid progeny, this will be called the F2 generation. And the F2 generation would be the worms that we actually looked at under the microscope. So in the F1 generation, we know that 100% of the worms are heterozygous, but they're heterozygous for different mutations. Although in this experiment we mostly focused on phenotypes, we still have to do the Punnett square to determine the phenotypes of the next generation. So since we know that 100% are heterozygous, by doing the Punnett square, we have come to this result that 50% of our worms will be heterozygous to the mutation, 25 will be mutated homozygous, and 25 will be wild type. So the low probability leaves us with a chance of 1 out of 16 worms with a desirable phenotype. In other words, if we're going to have a plate with 16 worms on it, only one will have the mutation that we're looking for. So when we were looking under the fluorescent microscope, the wild type will show this expression. This will be the DA9 expression, and what we were looking for is the mutated gene that would show beside the DA9 expression along the ventral nerve cord will show the expression of other neurons beside the DA9. So what we have done so far is 2 cycles of screening. We have screened over 4,100 F1 worms, which is the same as 8,200 haploid genome screen. We were using the ITR1 GFP fluorescent marker, and GFP stands for green fluorescent protein. And this GFP marker was used in order for us to be able to observe the ITR1 gene expression, for us to be able to see it. And so far, by doing the experimentation, we have found one possible mutant that could be the one that we're looking for, but we're still not sure because no one else have done this experiment before. So what we're doing right now, it is incubated at 20 degrees Celsius, and we're waiting for it to give progeny. So far, what we have actually looked at this mutant already, and we have realized that it's unable to actually lay eggs. And something that is going on is that the progeny is being growing inside of the mother. So we're waiting until they will be able to get out of the mother and actually live there on their own, and then we can look at the phenotype and see if that's exactly what we're looking for. So our future direction is just keep screening and hope for the best that we're actually going to find the mutant. And if you ask for a reason for all of this, why we're doing this experimentation, we want to discover new genes that are important for the mononeuron diversity. In other words, is that the genes that make one mononeuron different from another mononeuron. And also by discovering how each gene and how each mononeuron works in a simpler organism, we might discover a way of actually finding and looking at more complex organisms like mice or human beings. So I want to acknowledge Dr. Pascalis Gracious for being my mentor, for Dr. Albert Hopper for placing me with my mentor. Romulo Morado is an undergraduate student that has been working with me throughout the experimentation, the Harvard Lab for being welcoming, Dr. Sad, Harvard Society, and we guys. And questions?
When you talk about the gene ITR1, so what's this gene so special? We're not looking really why the gene is special. We're trying to figure out how the expression of the gene is different. Like, for example, the one gene can be expressed in one mononeuron, but it's not expressed in another. So what's so special about that mononeuron that it doesn't allow the expression of this gene?
Yeah. What happened when the gene was expressed? What difference did it make?
When the gene is expressed, because we were using the fluorescent marker, we would see something like this. We would see the expression of the fluorescent protein marker.
No, like behavioral changes, because you said it's you said it's a motor neuron, and you're changing part of the DNA by allowing it to express a gene that's not normally expressed. So, what's the side effects that it would
We
have?
don't know the function of the gene itself. That's why we're trying to figure out more mononeurons that will be expressing this gene, so we can actually take a closer look and determine how and what is it served for.